Introduction. Welcome to today's lecture on molecular biology. In this session, we will delve into the fascinating realm of chromosome organization and its role in development, disease, and various biological processes. Chromosome organization refers to the spatio-temporal arrangement of DNA molecules within the nucleus, which plays a crucial role in regulating gene expression, DNA replication, and genome stability. Now, let's explore the different aspects of chromosome organization and the intricate mechanisms underlying its formation and function. Chromosome territory formation involves the spatial organization of individual chromosomes in the nucleus without entanglement. This process is crucial for genome function and involves various nuclear structures such as the nuclear lamina, nuclear pore complexes, and chromatin-associated proteins. Scientific studies indicate that chromosome territories are primarily established during development and maintain a consistent pattern throughout a cell's lifespan. The loop attachment model proposes that chromatin loops attach to nuclear structures, segregating chromosomes into distinct territories. Understanding the 3D organization of the genome is crucial for gene regulation and stability. Disruptions in this organization can cause developmental disorders and diseases. Techniques like Hi-C have revolutionized our knowledge in this area. Chromosome organization occurs at different levels, such as topological domains and chromosome looping, which play important roles in gene expression. Architectural proteins, like CTCF and cohesin, shape the 3D structure of our genome. CTCF acts as an insulator, forming chromatin loops, while cohesin helps with DNA repair and sister chromatid cohesion. These proteins interact with DNA and control the communication between enhancers and promoters. Dysfunction of these proteins can lead to diseases. Chromosome conformation capture techniques, 3C, 4C, 5C, Hi-C, allow investigation of interactions between DNA regions. They identify physical contacts and reveal the 3D architecture of the genome. Each technique has unique advantages in studying genome organization. Summarize in 15 words. Explore chromosome looping and its role in gene regulation, including enhancer-promoter interactions and cellular identity maintenance. Uncover the complexity. Understanding how genes are regulated requires studying nucleosome positioning. Nucleosomes are DNA wrapped around histone proteins and form chromatin. Their placement affects gene accessibility to transcription factors and RNA polymerase. Nucleosome positioning changes dynamically due to factors like DNA sequence, methylation, histone modification, and chromatin remodeling. Alterations in nucleosome positions can impact gene expression and contribute to disease. Chromosome folding refers to the higher-order organization of chromatin, which influences gene regulation and genome stability. The folding of chromatin occurs in a hierarchical manner, encompassing different levels of genome organization. At the megabase scale, chromosomes form topological domains, which are self-interacting chromatin regions displaying distinct functional properties. These domains serve as regulatory units by facilitating enhancer-promoter communication and preventing the spread of heterochromatin into euchromatic regions. In cell division, chromosomes are organized and distributed to ensure accurate separation of genetic material. Proteins like condensins condense and hold chromosomes together. Studying these mechanisms helps us understand mitosis, meiosis, and genetic stability. Epigenetic modifications, such as DNA methylation and histone modifications, contribute to the regulation of chromosome structure and gene expression. These modifications act as heritable marks that influence chromatin accessibility, dynamics, and higher-order organization. DNA methylation patterns, established by DNA methyltransferases, can impact chromatin compaction and gene expression. Histone modifications, including acetylation, methylation, and phosphorylation, influence chromatin structure by altering the interactions between DNA and histone proteins. Alterations in 3D genome organization have been observed in various types of cancer and are considered crucial elements of oncogenesis. Disruptions in gene regulation due to changes in chromatin organization can lead to abnormal cell proliferation, the acquisition of drug resistance, and metastasis. Understanding the relationship between 3D genome organization and cancer can provide valuable insights into the underlying mechanisms of tumor genesis and potentially aid in the development of novel therapeutic strategies. Aging is associated with various changes in genome organization, including alterations in chromatin structure and gene expression patterns. These changes impact cellular functions, tissue homeostasis, and the aging process itself. Age-related changes in chromatin organization can result from cumulative DNA damage, epigenetic modifications, and changes in chromatin regulators. Investigating the links between chromosome organization and aging can provide valuable insights into the molecular basis of aging-related diseases and potential interventions. Disruptions in chromosome organization can give rise to human genetic disorders. Structural abnormalities, such as chromosomal rearrangements, translocations, and copy number variations, can impact gene expression, lead to gene dosage imbalances, and contribute to disease phenotypes. Examples of genetic disorders caused by alterations in chromosome organization include Down syndrome, Cre-Duchette syndrome, and various chromosomal rearrangements associated with cancer. Understanding the correlation between chromosome organization and genetic disorders is pivotal for diagnostics, prognosis, and potential therapeutic interventions. It is clear that the intricate mechanisms of chromosome organization play a pivotal role in gene regulation, DNA replication, and the stability of the genome. Not only is it vital for normal development and cellular balance, but disruptions in this organization can lead to diseases and genetic disorders. By understanding how chromosomes are spatially arranged and how they interact with architectural proteins and epigenetic modifications, we can gain valuable insights into the intricacies of cellular functioning.